Alrighty guys, I am going to go on ahead here and start on our second section. I just want to make sure that that is recording. It's been giving me problems lately. Okay, so we left off here talking about the bread a bit ago, and I told you that the holes in there were from the carbon dioxide that was being released as it went through alcoholic fermentation. So now we're going to talk about aerobic respiration. And I like this picture here because you can see this little guy. He has a plant on his back. And if you look at the world around him, it's all gray and cloudy and it looks kind of dismal and dark and there's nothing growing. So he's carrying this plant around him because that plant is giving him oxygen. It looks all nice and happy in there. There's like a little sun shining in there. Okay, so aerobic respiration has two main stages that only occur if oxygen is present. And just like in the first section, you're going to pay attention to the parts in red. That's what you're going to copy down. And that's what you're going to end up turning in after we finish these notes up. So there's the Krebs cycle. That's the first one. And it involves the completion of oxidation of glucose that began in glycolysis. NAD plus is going to be reduced to NADH, just like it is when we go through the fermentations. Wow. That is the Krebs cycle. Holy crap, it looks huge and confusing. We'll try to break it down as much as we can. The second stage is the electron transport chain. It's associated with chemiosmosis, which we talked about in photosynthesis. It's using energy released as protons to move across the cell membrane to make ATP. This is the stage of cellular respiration that produces the most ATP. And there's your electron transport chain. It occurs in the membrane of the mitochondria. So where did the Krebs cycle and the electron transport chain occur in the cell? In prokaryotes, it occurs in the cytosol. In eukaryotes, like us, it's going to occur in the mitochondria. The pyruvic acid produced in glycolysis diffuses across the double membrane of the mitochondrion and enters the mitochondrial matrix. So the mitochondrial matrix, this is a zoomed in version of it. There's little spaces in there and it's going to occur in these spaces, but this matrix is gigantic. It's basically like one big maze inside of here. And it's folded so that it can have even more space, more surface area to make ATP. When pyruvic acid enters the mitochondrial matrix, it reacts with a molecule called coenzyme A to form acetyl-CoA, abbreviated acetyl-coenzyme A. Acetyl-CoA has 2 carbon atoms, but pyruvic acid has 3. The carbon atom lost is released through carbon dioxide. So when you breathe out, you're getting rid of that one carbon dioxide. This reaction reduces NAD plus to NADH. The Krebs cycle is a biochemical pathway that breaks down acetyl-CoA, producing carbon dioxide, hydrogen atoms, and ATP. The Krebs cycle was identified by Hans Krebs, a German biochemist. Oops, that's my phone going off. Hold on one second, guys. Alrighty, guys, let's get back into it here. So we have step 2 here. Basically, we're regenerating coenzyme A. So that took care of that entire top portion up there. That is all step 1. Next step, we have the citric acid is going to be oxidized by releasing a hydrogen atom and a carbon dioxide molecule to form a 5-carbon compound. The electron in the hydrogen atom is transferred to NAD+, reducing it to NADH. That's this part right here. Step 3, the 5-carbon compound formed in step 2 also releases carbon dioxide molecule and hydrogen atom. This forms a 4-carbon compound. A molecule of NAD+ is again reduced to NADH. A molecule of ATP is synthesized from ADP. There's step 3. Step 4 is the 4-carbon compound formed in step 3 is going to release a hydrogen atom. The hydrogen atom reduces flavin adenine dinucleotide to FADH2. And there is step 4 down there at the bottom. In step 5, the 4-carbon compound formed in step 4 releases a hydrogen atom, regenerating the oxaloacetic acid to keep the Krebs cycle operating. The electron in the released hydrogen atom reduces the NAD plus to NADH, and there is step 5, and it's going to kind of go back into step 1 at that point. So the product of the Krebs cycle, you're going to get 6 NADH molecules, 2 FADH2 molecules, 4 CO2 molecules, 2 ATP molecules, which is the same amount of energy as glycolysis is producing. And then we're going to go into the electron transport chain and chemiosmosis. And chemiosmosis is where we produce the majority of our ATP. So it's a series of molecules in the membrane that transfer electrons from one molecule to another. That's what electron transport chain, that's what it means. So in eukaryotes, the ETC, and that's how I'm going to refer to it from now on instead of saying electron transport chain, and the enzyme ATP synthase are embedded in the inner membranes of the mitochondrion folds called the cristae. So that's what the mitochondria looks like if you cut it in half. In prokaryotes, the ETC is in the cell membrane. ATP is produced by the electron transport chain when NADH and FADH2 release hydrogen atoms, regenerating NAD plus and FAD. The first step, we're going to have NADH and FADH2. They're going to give up electrons to the electron transport chain. These molecules also give up their protons. Step 2, the electrons in step 1 are passed down the chain. As they move from molecule to molecule, they're going to lose energy. Step 3, the energy lost from the electrons is used to pump protons from the matrix, building a high concentration of protons between the inner and outer membranes. This creates a concentration gradient of protons across the inner membrane, meaning that there's more protons outside than inside. So we're on step 3 now. We're down in this area. Step 4, the concentration of protons drives the synthesis of ATP by chemiosmosis. This is the same process in photosynthesis. Just moving on down, and we're not hitting them with light this time. ATP synthase molecules are embedded in the inner membrane near the ETC molecules. As protons move through ATP synthase and down their concentration, ATP is made from ADP+. So that is going to be over here. And step 5, oxygen is going to act as the acceptor of protons from NADH and FADH2 and electrons that have passed down the chain. The oxygen, protons, and electrons all combine to form water molecules. The electron transport chain can produce up to 34 molecules of ATP. That's a lot of ATP. Remember before we were only getting like 2 molecules we could use, or we got 4 molecules we could use. 34 is a good bit. And that is that very last step down here at the bottom. It's showing you have oxygen plus these electrons plus hydrogen, and that's giving you water. The importance of oxygen. We know it's important, right? ATP can be synthesized by chemiosmosis only if electrons continue to move from molecule to molecule. In order for this to work, there must be a final electron acceptor, which is the oxygen. So without the oxygen, the chain is going to stop. And if this stops, then the ATP production will also stop. So this guy here, he's getting ready to serve a couple people a whole lot of ATP. Look at all those hot dogs. Makes me want a hot dog. In summary, a total of 38 ATP molecules are produced from each molecule of glucose. So we got our 2 from glycolysis, our 2 from the Krebs cycle, and around 34 from the ETC. Glycolysis occurs in the cytosol and converts glucose into pyruvic acid. A small amount of ATP and NADP plus is produced. Aerobic respiration occurs in the mitochondria and converts the pyruvic acid into carbon dioxide and water. And this produces a large amount of ATP. The overall cellular respiration equation is right there. Do your best when you write that. You don't have to worry so much about getting these subscripts in there. You could put a big 6. And the equation for cellular respiration is related to that of photosynthesis, in which the products of the cellular respiration are the reactants in photosynthesis. So I'm just having you copy it so you can see that they are just opposites of one another. And we're going to be making a foldable, if you're in class, based around this idea. The roles of cellular respiration is to provide ATP that all cells need to support the activities of life. facilitate the making of specific compounds not contained in the food that heterotrophs eat. For example, approximately 10 of the amino acids needed by the human body can be made from compounds formed in the Krebs cycle. So you're not going to get those amino acids by eating something. And that is it for section 2. So you will be turning in those notes to me, the ones you took from section 1 and the ones you took in section 2. Alrighty, guys. I will talk to you soon.